So today I would like to introduce to you BioNewmetrics, which is a website, a portal, where we disseminate several types of information, and this is the goal of today. So to introduce this portal, BioNewmetrics, and look specifically at the analysis of super resolution trajectories. So a few lines about what is this BioNewmetrics website. So you can have it at the following email address, www.bionewmetrics.org. So this initiatives was started in 2009, and the goal is really to present mathematical methods applied in various fields of life sciences. So what you will find there are modeling, so how we model different parts of the cell from a molecular level, numerical simulation, specifically stochastic simulation, how we do data analysis, and we have now example about phototransduction. And this website is also about education. We have several lectures that are available on YouTube, and these educational lectures are about mathematical modeling at various levels, at the master level. So the theme of BioNewmetrics, so we present various themes of analysis. So as we already mentioned about phototransduction, but more generally, we have also tools to analyze electrophysiological data. Today, I'm going to present a specific analysis about super resolution trajectory. Super resolution is a new way to break the diffraction limit and thus to get refined trajectories. And the question there would be, how can we extract data about the biophysics underlying those trajectories? Pretty soon we will have a polymer simulation, and this is important to have interpretation of chromosomal capture data, and most generally how the nucleus is organized. And we're currently working on voltage dice dynamics analysis. So we have already published a paper about how is it possible to make numerical simulation in empirical domains. And so pretty soon we will also post in the BioNewmetrics website this associated algorithm. We'll have also baby cries. We have a database and how to interpret the cries of your baby, and also a cohort of heart monitoring data. And this would be important, especially to design new iPhone applications. So if you log into BioNewmetrics, this is basically the first page that you will find. There are several options for open lecture, the project. We explain who we are and why we did this, and you have a possibility to contact us. Now if you click on one of these elements, you are directly, so by clicking in here, this is a short description, for example, of this super resolution single particle trajectory using stochastic analysis. So again, if you click in here, what you get is you get to another page where we have here a little description of what this page is about. And you have here three elements. One is a description in the PDF format about what this application will do, and you have here the entire Matlab code that you can use for yourself. This is a GUI for the application, and we have an example of data that if you don't have your personal data, you can just use this as an example. So if you look specifically on this data, you get here, for example, here it is about the particle position. So I guess we have X, Y, and other element of information about the position and time of the particle. And then this is the GUI that after you run the Matlab application, you get this window. And so basically you can load your file by clicking here on the format that is appropriate. You can also load an image. So this is an example of an image of the dendrite of a neuron. And then you can here specify some information about the resolution in space, the resolution in time, the information about the frame, the trajectory identity. And then we have already pre-calculated a certain number of features, such as the probability density function, the diffusion, and the drift. So this is an example of if you have many trajectory and if you superimpose them on a dendrite, what it gives. You can calculate the diffusion tensor or diffusion map at each point, which give you the intensity of diffusion locally, and you can also calculate the drift. So the goal now of this short presentation is to explain through this example what we do at BioNewmetrics. And so first of all, I would like to introduce to you basic concept about stochastic theory. Then I'll move on to the description of the methods to extract biophysical information, biophysical parameter and features from the super resolution data. And then give some application to different data that is available on the market, such as AMPAR receptor, NMDA receptor, glycine receptor, calcium voltage channel, and also other type of systems such as the GEG protein that are important for the formation of capsid assembly. And then I would like to discuss how we can use this information extracted from data to make simulations in empirical domain and describe briefly what is the procedure. There will be other videos to explain this in more detail. So let's go to the general stochastic theory. So basically, if you're interested in extracting information from many particle trajectory, what you need is an underlying model. So we start with the Langevin model, which is basically Newton's law of motion, where noise has been added, and we have discussed this in many different classes. So you have acceleration plus the friction term plus force equals Brownian noise, and in the Smolov-Hofsky limit of the Langevin equation, when gamma goes to infinity, you can show that in probability this first differential equation converges toward this what is called the Smolov-Hofsky limit, where you have the position, the force, and the term about the Brownian motion. So in conclusion, we have a model here about continuous stochastic particle trajectories. So more generally, you can look for a physical description of trajectories as the velocity, which is generated by force, and the force is usually computed from first order moment statistics around the trajectory, and then there is a noise term which is usually calculated from the second moment. Note that this coarse-grained description does not necessarily satisfy the fluctuation dissipation theorem of statistical physics. So B of x is a drift, which accounts for local interacting potential, and D of x is the effective diffusion coefficient that accounts for crowding, but not necessarily not for binding, as it is already contained in the drift term. So now that we have a model for our trajectory, our goal will be to find from trajectory, to identify can we compute or estimate the drift B of x and the diffusion coefficient D of x. And so the goal is really to do a reverse engineering problem to invert this equation from trajectories. And so the solution of this problem is well known. This is already contained in Feller's work, and it goes like this. The drift B of x can be calculated from the limit as delta t goes to 0 of the quotient x of t plus delta t minus x of t divided by delta t, condition that x of t equals x. And then we have to take the expectation that is the average over many realization. So from this you can use the empirical sum, which is the following. And this is discussed in other lecture. It turned out that this empirical sum, which is a non-parametric approximation, is non-biased, and it is also the best estimator, best in some sense that I'm not going to enter here. So in other words, this inversion procedure requires many trajectory passing through x, as we need here, as we see from the expectation, we need to average. And in practice, it is important to calibrate the size of the mesh, such as n is of the order of 100, 200. We have estimation of this in the paper of Oze et al. Published in PNAS 2012. So we have the same for the diffusion tensor. This can be calculated similarly using the following empirical sum. And once we have recovered the drift and the sigma of x, basically we have everything to simulate or to characterize our trajectory, because those are the element of the stochastic equation. So we can reconstruct the dynamics, we can estimate the probability density function or any transition from any region of interest, such as dendritic spines in the case of neuronal cells. And we can also calculate mean first passage time, which is the time to go from one part to another, but this requires usually to add external boundary conditions that are not contained in the biological features. So now let's go to the second part, which is description of the methods and the application to the data. So our colleague from Bordeaux have given us the opportunity to analyze their trajectories. So this is trajectories coming from super resolution data. So you can have tens of thousands of these trajectories. And the question is, what is the mechanisms responsible for generating those trajectories? So basically they are acquired at 50 milliseconds time interval, and in average they are about 7 consecutive points, and after the trajectory stop to exist as physical trajectories observable. So when you superimposed on a single cell all the trajectory, you get this beautiful picture. And so if you magnify region 1, 2, and 3 there, what you can see, there are region of high density, which is labeled here in red. And so a fundamental question is why there are regions where trajectory are accumulated compared to other regions. What is the reason for this heterogeneity? So this is the question that we have partially answered in this publication, and so now I would like to explain the methods for this. So we use now the estimator for the drift that we just discussed a few slides ago, and now we will do this reverse engineering problem by estimating B of x. So you take a grid. So you see that in green you have the trajectories of different trajectories, and then when you arrive to here, we do the averaging. So we calculate this b of x for each pixel of our grid. And then you end up here with a map of vector fields. And now the difficulty is to what can be extracted from this map. So you can see in here that we have a region here in red. I'm sorry, that I just now mark in red, but those are the white region here. The white region, which is the marker of synapses, and you can see that on this region there is really a specific behavior of the field, where the field seems to the amplitude of the arrows are vanishing, and so we are interested in understanding what is the reason for this. So before continuing on this, the analysis allowed us to look at the map of diffusion coefficient. So each color here, the color code is given here, gives you at each point the local diffusion coefficient calculated from the estimator that we presented above. So this explains the GUI that is presented in BioNewmetrics. So not only you have here the initial figure, we have the density, you have the diffusion coefficient and the drift that we calculated through the procedure of averaging and the empirical estimator that we gave. So now let's discuss the application of these methods to the super resolution data and to explain what is the meaning of the converging arrows. So how to find features in the vector fields. So at this point it is important to note that conservative force in physics are given as a gradient of a potential. And classical potential well in dimension 1 can be represented generically as a parabola, and in dimension 2 you have this paraboloid, and if you take the gradient of this paraboloid, you get these converging arrows, which is a signature of a potential well, which is a clear mark of a local interaction. So note that this is now an in vivo interaction, something that we extract from in vivo data. So what we did is we looked in region where actually we could see potential wells and converging arrows, and we did found them, some of them. So to make sure that this was not due an artifact to the methods, we looked at how these arrows were changing across different scale by changing here the radius of the of our grid. And what is important here to note is that although the size of the arrow is changing, they are still converging to the same spot, showing that our result was independent of the radius r of the analysis, confirming that those regions are clearly given by potential wells. And so to confirm this, we obtain different potential wells at different locations. So this is coming from, we calculated here the wells from empirical data, and this is obtained by fitting here this parabolic approximation on the empirical data. And you can see the similarity here between the different wells and their reconstruction and the empirical data, confirming that this approximation by a parabola was clearly very good. And in this paper in 2012, we gave an index about the likelihood of having a potential well. So to summarize what we have done here, you can feed an image, a confocal image. Then you can, once you get the information about your super resolution trajectory, you can use the algorithm that we presented to have this vector field and to extract this here in red, this region of interest where you have converging arrow, which turn out to be synapses located at the dendritic spines. So you can see that the method can be applicable on a different type of super resolution data. So this is an example of calcium channel trajectories, and so the difficulty was to give an interpretation for the region of confinement of the trajectory. And we found that they are due to a potential well, which is a signature of interaction, and we could extract here the energy of the wells. Another example comes from the GAG aggregation protein. This is in collaboration with our colleague from EPFL. And again, we could identify the region where you have this hotspot here labeled in 1, 2, 3, and 4. And we found again that they are due to converging arrows, which is a signature of potential wells. And we extracted here the energy of those potential wells, as well as the size of this region of interaction. So the method is quite general and can be applied to different types of data. And I would like to conclude with a possibility to use this data using numerical simulation to extract additional information about the biophysics underlying these dynamics. And so this is about simulation in empirical domain. So once we have this vector field on the image and the stochastic equation that we presented, which is a Smolov-Hofsky limit of the Langevin equation, we can really use this to generate simulations as shown in here. In blue, this is a simulation of a trajectory inside the image of the neuron, inside the confocal image. So there are various technical difficulties here that I'm not going into the details, but basically you can generate trajectory starting at the point A and ending here in the green circle, and do and run many simulations so you get statistics about your trajectories. And so you can now calculate various quantity of interest. So in here we summarize the algorithm, which is presented in 2014 Biophysical Journal paper, where we have this initialization procedure, then a procedure that where we can extract, so the initiation procedure where we extracted, as we discuss here, the drift and the diffusion coefficient. From this you can have an algorithm to detect the potential well and to identify your vector field in the domain of interest. There is also a procedure here that I'm not going to discuss much, is to be able to run a spatial filtering, low-pass filtering, so that some of the pixels which are coming from your coarse-grained image of confocal images should disappear, because once you run your simulation, they are coming from super resolution data, which are at a much better high resolution scale. So we have to be careful not to introduce and not to mix information from different scales. So this is an example of trajectories that are run on the image, and of course we can run them with no drift or with drift. And then, as we have shown from the narrow escape theory, the mean time it takes to escape from a dendritic spine is Poissonian. So we can show this, you can recover this property from the statistics. And then the only parameter that needs to be fitted here is the lambda, which is your parameter of the exponential, and 1 over lambda is the mean residence time. So this gives new type of information that's allowed to calculate residence time of any quantity of interest. This we gave the example of a dendritic spine, but it can be in any shape on any cell surface. So to conclude this introduction of BioNewmetrics through this example of super resolution data analysis and simulation, this is a platform to provide software analysis. We also provide a simulation toolbox for phototransduction, how you simulate the activation of a cell, such as photoreceptor, by a single photon. We have also mathematical modeling, narrow escape, about the narrow escape theory, which is the theory to calculate the time for a Brownian particle to find a small hole on a closed domain. We have also educational different classes available on YouTube, but they are dedicated to modeling and applied mathematics and asymptotics for problems in molecular and cellular biology. And finally, there are now several conferences related to this field of mathematical modeling, statistical physics, for biophysics and biology at the molecular scale. The future project includes models and simulation about polymer physics for nuclear organization. So I'll stop here, and I wanted to thanks all people in the group and collaborators that have been providing us data over the years.